every system has some advantage and disadvantage because it's a naturally occurring transmembrane protein. Our system's advantage is that the heavy role of delivery to the cell is done by the transmembrane protein. Our particles get close to the cell membrane. It interacts with the cell membrane, allow our particles to fuse. A particle itself becomes a part of the cell membrane and the cargo directly go to the cytosol, which is different from the LNPs where it mostly end up endosomes. And then you have an endosomal escape, which is not the case in our part of delivery.
Gene therapy only works if the cargo reaches the right cells intact. AAV and lipid nanoparticles have carried the field this far. But a different class of delivery vehicle is shaping what's possible. Today, Jitendra Kumar, who is the lead scientist for chemistry and process development at Entos Pharmaceuticals, joins us to unpack proteolipid vehicles and diffusogenic platform, and why the distinction matters for manufacturability. Let's get into it. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Jitendra. It's great to have you on today.
Thank you, David, for the invitation.
Very welcome. To get us started, let's start with this perhaps a bit controversial question. Share something that you believe about bioprocess development that most people disagree with.
In my opinion, bioprocess development, especially coming with the new technologies where like LNPs and AV are, it's very complex and it involves a lot of structural biology informations as well, which is a background about the development of these technology. Even though in terms of the methodology development, you see a straightforward pathway, but there is a lot of background to it.
Yes, and I'm excited to dive further into this world during our conversation. Tell us a bit more about yourself. You have quite an impressive career, a long career. What sparked your interest in science, and what were some interesting pit stops along the way that led you to your current role?
First thing is I grew up in a family of scientists. My father was an agronomist in an agriculture university in Frankfurt, India. And obviously, sitting around the people who are in the science, it was a natural development in the science. And during my journey, I met a lot of people who encouraged that development further. My natural inclination was towards the physics. And that is where I got more into the physical chemistry side of the things, physical chemistry, analytical chemistry. And that is where I enjoy most. In terms of the current role, I did my PhD in Goethe University, one of the biggest NMR center in the world, with Professor Harald Solvay. It's amazing to work with him and getting the experience, which is in academic setup, you get an industrial experience. The way the whole lab work is pretty much like an industry. We have a very defined objectives there. And current role was a transition from the structural biology to getting into the industrial role where I was filling a space where they needed more people into the field. And I got into this role, I will say, being lucky because I had a good network of the people who supported me in finding this role. And the one thing which I will say to anybody is that always enhance your network because you never know how the opportunity will come to your door. The answer to your question is that science is a natural inclination because of my family environment.
I agree that your network is important. And that's something I stress also when I talk to younger scientists. Perhaps I'm curious, you had this perspective too as a younger scientist in your career. You think science is the most important thing? Yes, science is important. However, building relationship is equally important.
Yeah, it is very important. And I worked with almost 15 years working on a prion protein, which is one of the most difficult protein to work with. I worked on a structural biology of it in a Herald's lab in Frankfurt, and then I got more curious about the biology of it. So I moved to Edmonton, Alberta, where I joined in a prion center with a neurologist as a postdoc. Even still working on biophysical techniques, but on the other hand, I got exposure to the more biological perspective of the disease and working with a person who actually deal with the patients, seeing the patients who are getting affected with the disease. It brings you a different level of knowledge, and not only the knowledge, the impact the disease is having. And I still remember the days when I'm in the lab and my supervisor at Valerie Sim coming from the hospital, and you can see on her face that she met somebody who actually diagnosed with the disease. And being a doctor, you know that there is a certain timeline you are giving to that person. And that gives a lot of motivation for the research, because what you are doing is actually you are trying to impact the life of those people in the future. It's a lot of encouragement, but also the sad reality of the life. There are a lot of rare diseases, a lot of things. We still face a lot of big challenges.
Yes, absolutely. And I love that your science and your work is driven by a big purpose. Let's talk a bit about that, because you and your company, you want to deliver on the promise of genetic medicines. So tell us what that means and also how you go about that. What is the technology or the solution you're providing?
Coming back to the company, Entos Pharmaceuticals from University of Alberta. It's a startup from the Department of Oncology here. We studied in oncology sector. We're trying to silence few genes or expression of few genes. The thing is, genetic medicine is pretty much a future for many of our diseases. And if you look into the broad perspective, you can find a lot of diseases either as a loss of function or the gain of the function disease. In either case, having a small molecule which can deliver the job is good, but if it's a gain of a function where you are having a protein expression, overexpression happening, applying a siRNA or ASO, we can suppress the expression much effectively, is a good technique. Similarly, where it's a loss of function, you can introduce a foreign gene or natural gene, different drug delivery method to express. Is going to give you a natural desired expression of that protein, which in many cases we can do. Say, for example, antibody therapies are there where you are introducing antibody, which is a foreign protein you are giving as a dose. But genetic engineering is promising in a way that you can regulate it. It's a multi-step process there. So regulation can be done with the promoters, enhancer designs. So there are multi-factors there where you can, with the genetic engineering, have a controlled treatment. It's still a little futuristic approach, I will say, but we are getting there. Coming back to the company's role here, we are close to the LNP technology if you compare non-viral therapy. But on the other hand, I will say it's an amalgam of actually two fields where we have a viral and non-viral together. We have identified a small transmembrane protein, which is called a fusion-associated small transmembrane protein, which is fast protein. And it's a family of about 10 proteins identified by Roy Duncan in Technology University. And then in his lab, they have created chimeras, and the proteins which we work with is one of the chimera, which we have a proprietary on it. And it is one of the smallest transmembrane protein known. And it allows our particles to fuse directly to the cell membrane, which is a major bottleneck for many of the technologies in the field for the drug delivery.
So that means that, as I'm paraphrasing this, you developed a novel delivery mechanism to deliver whatever gene or whatever substance you want to deliver to the site of interest. Perhaps to draw a picture, when we look at gene therapy, usually you use kind of a delivery vehicle like AAV, for instance, or others to transport. So how is this mechanism different, and why is it better, or what are the certain advantages versus more traditional systems?
I will say that every system has some advantage and disadvantage, and I will talk about the advantage of our system. And because it's a naturally occurring transmembrane protein, which is having a functional role of fusing with the cell membrane. Our system's advantage is that the heavy role of delivery to the cell is done by the transmembrane protein. And that is the bigger task in a lot of delivery vehicles. So what is happening, our particles get close to the cell membrane. It interacts with the cell membrane, allow our particles to fuse. A particle itself becomes a part of the cell membrane, and the cargo directly go to the cytosol, which is different from the LNPs, where it mostly end up endosomes, which is like engulfing inside, and then you have an endosomal escape, which is not the case in our part of delivery. And the advantage which compared to the LNP space caves we have here is that we are not dependent on LNPs or lipids for the delivery. We have a much broader space of lipid flexibility. We can change the lipids much more easily, and that allows us to address the tropism.
And what are the difficulties you're encountering with your platform?
I will not say the difficulties, but like if I compare with the LNP at the moment, let's put it in this way. We have a protein component. So if you talk about the CMC, we introduce another factor, which is biological. So rather than having a straight chemistry-based CMC here, we have a biological process where we are purifying the protein, and that protein becomes a part of our vehicle. Rather than the difficulty, but it's a slightly complex, the traditional LNP in that sense.
So what's difficult or challenging with your platform because you're using a biologics production system to get your delivery vehicle?
It's different than the LNPs. Right now, our process is pretty much optimized. We have no challenges as such, but it's a pretty much like a monoclonal antibody or any protein production. You have to follow the same path for the production of this fast protein. In terms of the challenges which I faced initially, there is practically no information available in a regulatory database for the membrane protein. Everybody talks about the soluble globular proteins or monoclonal antibodies, but membrane protein space is pretty much new for the regulatory.
But you're producing this recombinantly as well as you would an antibody or other molecules.
Yes, we are using a baculovirus expression system for the expression of the protein, and that allows the post-translation modification on the protein, which is required.
And this system would also then enable easy scale up, for instance, if you need a lot more of this molecule.
We have done a few scale-up studies, and I just want to add here is that we have a facility in Carlsbad, California, where we can do the phase 1, phase 2 productions. We are pretty much in a good space for that kind of production at the moment. And we have done a couple of studies with the support of Cytiva for the scale-up, which work pretty well. Having said that, it's a membrane protein, and anybody who worked with the membrane protein or in a space knows that there are inherent problems associated with the membrane protein. So it's one of the issues which I can say at the moment is there is a limitation of the expression. It's not a highly overexpressing protein. So the scale-up is very linear than exponential, and we are working on it. There are few things here and there we are trying, and we are having a promises, and that's where it was great to work with the Cytiva, where they provided the bioreactor for the 50 liter, and we did a pretty good run with them.
Excellent. And tell us where you are in this development. Are you already in clinical studies with that? Is this still a proof of concept? How easy is it to get this to other people to use?
It's a very straightforward to use, and as a company, we are offering that to people who are interested in. Coming back to actually my journey, I will say that the time when I joined the company was that we got the approval to do the phase 1 trial with the COVID vaccine at that time. And that was the time when company needed more people, and I had a good transition happening at that point. So we are a clinical stage company. We have done the phase 1, phase 2 trials for the COVID vaccine in Canada. So in terms of the technology, we have proven that it's a clinically safe technology. It can be utilized. The question is, when it comes to the different targets, we still have to test out for the different targets, or if we are changing any lipid compositions, then we might have to have a little bit change there. But in terms of the core technology, we have proven that it's clinically safe.
And what kind of applications are you focusing on with this technology?
There are multiples at the moment. We are working very focused and a lot of lung diseases at the moment. I can say that is where current excitement in our company is hitting. When I'm saying hitting, we are delivering selectively to the lung, which is a challenge for a lot of LNPs, especially escaping the liver and delivering to the lung. We are pretty good there. And one of the key factor with our technology is that since our delivery is based on fast protein, there is no toxicity associated relative to the LNPs, which mostly goes to the liver. Those are considered as a liver tropic systems. And that is advantageous for certain things, but not advantageous for the everything. And that is where I'm saying that there is a space for almost every technology to be there, because everything brings some advantage. And so current focus is on the lung. And then we are also working on a disease called lipodystrophy. We are doing a leptin therapy. So currently there is a peptide-based therapy which people have to take daily. And our animal model studies are showing that so we can have a single dose which can restore the expression of the leptin for 6 months. And this is something which we are hoping to take it to the clinic soon.
And I'd be curious about the strategy when you're designing these phase 1 or phase 2 clinical studies. I mean, you need to look at the manufacturing. What are some key decisions you lock in early and perhaps things that can wait later?
To answer your question, when I got involved in the company, we were already starting the phase 1. And the thing which I learned, especially that was a difficult time for any company, especially for the startups during the COVID period. There were a limited supplies, and we as a company got lucky that there is a small scale production facility within the University of Alberta. So that is the facility which we use for the phase 1 production. And because of the limitations to reach out to the other people. It was a very good learning curve for us. Almost all the company staff at that point became GMP trained people as well. We changed our hat. And based on that experience and interaction with the people at that point, I feel one of the big challenge for the small company is a technology transfer. So for the phase one, if you are a startup, my advice will be you have to find somebody who understand and who get on your page easily, then going for a big CDMO where transfer of technology takes a lot of time. And here during that experience, what we saw is that's because we were the people who got trained to do the production ourselves. So speed up the whole process. Timelines which we were looking beyond 1 year, we did in 3 months. And that's a significant. And that particular experience actually led us to have our phase 1, phase 2 production facility in Carlsbad, because we realized that if we want to get into the clinic fast, that's the way to go. And we are open to support any other company for that perspective as well.
Speaking of small companies, I mean, you often have to make a trade-off between process robustness and speed. What is your take on that?
I mean, there is no trade-off for the process robustness. The process has to be robust. It comes down to how many checkpoints you have. And the one key thing which I tell my team is that identify the pause steps where you can halt. So right from the beginning, I came from academics. There is a transition, and we have to understand then when we are doing these, especially for the production space, finding out the right people. And to be honest, in that regard, as a company, we were lucky to find the people who were directing us in the right direction and in a speedy manner. And also finding the partners, as I said, ACTM here in University of Alberta, they were very supportive. So I will say that we were lucky, but also that's where your network comes, how you finding out the partners. And there are a few people we approached and they were big names and they did not work out our timelines. But it's a balance. You cannot escape the process robustness. Your process has to be robust. You have to be confident on your process. But on the other hand, if you have a technology and you want to make an impact, you want to hit the clinic as soon as possible. So speed is also a key factor.
I agree. The process must be robust, no doubt about that. The question sometimes I see is, okay, what are the nice to haves and what can wait until later? That's often a question small companies ask.
Yes, and that is where our head of the CMC, our head of the manufacturing is pretty experienced person. He has over 30 years of experience, and we were lucky to have him on the team. And those experience counts a lot because he was able to guide us what is must, what can still address later. So identifying those things as early as possible is pretty good. And other thing, which in terms of the production cycle itself, as I said earlier, finding out the pause steps, because whatever we think, we cannot have a continuous process. There will be chances for whether it's a mechanical failure, any other issues that we have to pause a process for some reasons. And if you have a process where you have not identified, where you can stop and restore and restart again, going to be a problem.
Yeah, absolutely. Delivery is where genetic medicines succeed or fail. Jitendra Kumar has already given us a lot to think about, from what sets proteolipid vehicles apart to the realities of working with fusogenic proteins. There is much more ground to cover in part 2, including production, characterization, and the CMC decisions that shape a novel delivery platform. Thank you for tuning in today. If you're enjoying the show, please leave a review on Apple Podcasts or your preferred platform. See you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.